You're listening to an Airwave Media Podcast.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Jess Steier
and Dr. Sarah Scheinman.
So today is kind of an interesting episode. We're going to dive into the world of med tech. And this was all spurred by this social media post that we did back in May. The tagline of that post, which I'll share to our show notes, is, "Can AI really mimic the immune system?" And now the inspiration for this post was this article, which I'll also link to our show notes, which described this incredible project, the Human Immunome Project. So I'm going to read just a little excerpt from our social media post, and then we're going to introduce two very special guests who can actually help dig into this topic. So here is the caption. Fun fact, we have explored and understand more about oceans, 5%, than human immune system function, less than 1%. However, the scientists at the Human Immunome Project, HIP, are looking to change that by taking on a massive project. They aim to capture the diversity of immune systems that are out there. This way, doctors, pharmaceutical companies, and medical systems will get a better picture of all the unknown factors that influence how easily someone might get infected or how their body will react to medications and immunizations. The overall aim of this project is to create a database that will aid in creating predictive models that can then be used in diagnosing and treating people. Sounds pretty interesting, huh? So today we are joined by two members of the HIP team. I'll introduce Jane Metcalf first. Jane is the co-founder and former president of Wired magazine. More recently, she founded and served as CEO of Protolife, a digital media company covering the neo-biological revolution, how biology and technology come together to impact health and well-being. No big deal. She is the board chair of the global nonprofit scientific initiative, the Human Immunome Project. And we're also joined by Dr. Hans Kiersted. Hans is a neuroscientist, world-renowned stem cell expert, and serial entrepreneur. He's the CEO of the Human Immunome Project. In addition to his role at HIP, he is the founder and chairman of Immunis and the founder and chairman and CEO of Avita Biomedical. Phew! How do you follow something like that? Hi, Jane and Hans. Thanks so much for joining us.
Great to meet you. Thanks for having us.
So can you, either of you just talk us through a little bit, like what exactly is the Human Immunome Project?
I'll take a stab at that, Jane. So it is genetics, but it's a lot more than genetics. It's very important to note that only about 10 to 30% of your health span or lifespan is dictated by genetics. 70 to 90% of your health span and your lifespan is dictated by how those genetics are turned on and turned off. And so if you internalize that for a moment, the next question makes itself very apparent. What is responsible for turning genes on and off? The number one thing in humans that does that is your immune system. So the immune system is extraordinarily complex and highly redundant, and it modulates what genetics are turned on and off. So the immunome is far more than genetics. It is also the cytokines and chemokines and growth factors, those little lipid or proteinaceous molecules that float through your blood, through your tissues, and influence your immune system, telling it what to do. It is the rolling of the immune system in and out of the blood to surveil and take away garbage and kill bad stuff, heal. It is the interaction of the immune system with every system in your body. Put simply, the immune system is the epicenter of human health. It determines whether we survive infancy, what diseases we get, how we age, how long we live. It's really as close as you can get to your health.
So I have a slightly different answer, because that's sort of the medical life science answer. I've got like the Silicon Valley answer. So you basically have a tech stack, right? So you start with the genome, but your immunome includes your genome, your transcriptome, your epigenome, your lipidome, your proteome, your microbiome, your metabolome. All of that omics kind of thinking is systems biology. And our artificial intelligence and machine learning is giving us this opportunity to think about all of these things more dimensionally than we have in the past. So all of those things make up your immunome, but then they're also influenced by diet, pollution, stress, and other externalities, which we call the exposome. And so the Human Immunome Project is an attempt to map and model the human immune system as it also then responds. So there's like your baseline, but then there's how does it respond to threats? How does it respond to a vaccine or a pathogen or, stress or poor diet or what have you? So all of those elements will go into understanding how the way Sarah responds differs from the way Jess responds. Why some people have cancer cells that never proliferate. And in other people, they start to reproduce out of control. And in still other people, the cancer will metastasize. Or why some people get COVID and it's a 2-day thing and then they're back at work. And other people, get really sick. And then yet other people have long COVID. Mm-hmm. So long COVID? Oh no,
not long COVID, but I raised my hand if you're just listening to the audio when you were talking about getting super sick from COVID, because it ran through my household. My husband had a sniffle. He wasn't even going to test. And I'm on, completely laid out for a week with a high fever, feeling super sick. So it's like, exactly. How is it that we responded so differently to the same virus? Sorry, I didn't mean to interrupt you, Jane.
Well, no, I mean, COVID's a really great example of why this is so important. And, we repositioned our precursor organization to what we are doing now during the pandemic because it changed the way everybody's thinking about, your immune system. Mm-hmm. And, I'm noticing this summer that people who are getting COVID are getting a lot sicker because the variant, I think it's the K2 variant just seems to be hitting a lot of people who are, vaccinated and have built up lots of antibodies and have been able to, bounce back quickly from previous infections. And they're having a harder time with this one, and for the neuroscientists here, it's also like neurodegenerative disorders, right? Why do some people die with a brain full of plaques and tangles who never had dementia? And other people, start suffering at an early age. So trying to understand that really gets at the whole root of human health and disease.
It feels like we're witnessing history. Honestly, I mean, this is really incredible. Jane and Hans, thank you so much for joining us today. Sarah, I know you're going to have a million questions for the two scientific officers, so we'll gear up for the second half of this episode that will tackle that. Again, Jane and Hans, thank you so much for your time and for everything you're doing to revolutionize medicine and healthcare. So thank you so much.
Of course, it's a fun ride.
Thank you guys so much for having us.
All right. So we spoke previously with Hans and Jane about the business side of the Human Immunome Project. And today we are joined by two more guests who I'll introduce in just one moment, but they are the co-chief science officers of HIP. So let's go ahead and introduce our two guests. We are joined by Dr. John Sang. John is a systems immunologist and computational biologist. I'm going to need to understand what those things are, but moving on. He is a professor of immunology and biomedical engineering at Yale University and the founding director of the Yale Center for Systems and Engineering Immunology. He additionally serves as an executive committee member of the Chan Zuckerberg BioHub, New York. We're also joined by Dr. Shai Shen-Or. Shai is an expert in systems and computational biology, informatics, and immunology. He's an associate professor at Technion, Israel Institute of Technology, the director of Tech AI Biomed, and the Zimin Institute for AI Solutions in Healthcare, and co-founder and chief scientist at CytoReason. So we have two, super lazy, not ambitious.
How do you have time for the Human Immunome Project is my question. Like such a slacker.
Yeah.
I do like two things.
Yeah, seriously.
We have genetic mutations that allow us to sleep for a long time.
Oh, I wish.
So we were thinking, so again, we talked about the business side of things. Now we really want to get into the science of it all. And Sarah had a really cool idea that maybe we could structure this conversation as sort of like a scientific research paper where we can get into the intro, the methods, the results, and the discussion. So Sarah, I feel like this is a little bit of a journal club. Do you want to kick off the introduction?
That's what I was thinking, because I just came from 5 days at the Society for Neuroscience conference. Just 22,000 brain scientists, aka my personal paradise. And I had the privilege of talking to like a bunch of different people that do research that's very different from my own. And I just found that the best way to structure that discussion was to tackle the why, how, what, and where of the research, and that also known as the introduction, the methods, the results, and the discussion. So if that sounds good to you guys, I think we'll start at the perfect place to start, which is the introduction. And when I teach undergraduate students how to read papers, I tell them that the introduction of a paper or a research study is meant to answer the question why, and to establish kind of the purpose of the study and any relevant background information. So I guess the first question that I have is, what exactly is an immunome?
We wondered that ourselves for a while.
Okay, perfect.
May I ask you a question back first, actually?
Let's dig
What
in.
is the genome to you?
Right. As I understand, the genome is the mapping of the chemical base pairs, the A's, T's, C's, and G's, to specific genes in the human genetic makeup.
Right. So if you apply that analogy to the human immune system, what do we want to know about our human immune system? Everything, right? So everything that we want to know about the human immune system would be, I guess, a part of the human immunome at the individual level. Like each one of us would have a human immunome that we carry. But the major difference between that and the genome is that the genome is fairly static. It's a static book, right? While the immune system it's capable of change and capable to adapt. Therefore, we need to understand not only where we are now, but we also need to know how it's evolving, how it's changing over time.
And maybe I'll add to that it's not just capable of adapting, that's its whole purpose. This is a sensory system that's basically, kind of continuously monitoring both the external environment and the internal one and responding to it, right? So just by living life, your immune system changes, and understanding that is key to actually, just given, and I guess if you add to that is that this is central, this is the system that responds to diseases and pathogens. So understanding that variation, how the baseline relation of how the immune system differs between all of us, and how that relates to changes that are happening environment is key to actually resolving diseases and getting treatments to work better, and ultimately be able to assess, what we call immune health. I like to say it's as if somebody walks to a doctor's office and says, "Hey doc, how's my immune system doing?" And it sounds like obviously we all get that it's an important question, but we don't have a good answer for this, despite the fact that we recognize it's important. And if you compare it to something like the heart, cardiovascular system, the doctor has a very good answer.
The other aspect that Shai just mentioned that's critically important, it's the connection between the immune system and all of human physiology, right? So it's this system that is not residing in one location only in your body. It's not a specific organ, so to speak. We call that a system because it's a distributed network of cells that surveys your body and monitors what's going on. As Shai mentioned, both external and internal stresses. And in a way, if you think of something called a buffer, are you familiar with the concept of a buffer? So just a couple of days ago, I had a nice discussion with Hal Barron, actually, who's the CEO of this company called Altos. He was talking about buffering in biology, and the immune system, in a way, is like the ultimate buffer in a way. So you may have disturbances and stresses and all of that. The immune system basically responds to these to make sure that these stresses are dealt with, and then we're going back to homeostasis, basically.
Oh, I love that.
Yeah.
The immune system is a buffer. I've never heard that before, but that's such a perfect analogy.
We love an analogy.
even just to get this to a to real life scenario. You mentioned, Sarah, you went to a conference. If somebody goes and gives a talk or gets interviewed, the immune system responds to this. You can see it in the data, right? Your cells change. There's here's another stress. The lion is chasing me. The modern day lion. Go give a talk.
Can I ask? And I'm very far from the biomedical world, so this might be a very stupid question. But so is the goal to take samples from people around the world and then develop some sort of a is the repository database? I'm not sure what the right word is of the immunome. I guess I'm curious because we're discussing how it is fluid, right? It changes constantly. Are we taking? I mean, is the goal a cross section? Like at one point in time, we're taking these samples. Is it longitudinal? Can you just speak?
Now we're getting into the how.
That's the how.
Yes.
No, that's perfect.
Okay.
That's
Are we ready for the how?
I think we're ready.
Okay. I think maybe let's keep the analogy to the genome again, right? Let's keep the analogy sort of parallel to the genome. When you look at the genome, we have been, meaning we as in the broad scientific community, has been trying to understand what information is contained in the genome and how does that information specify health and disease, right? So you've heard of genome association studies, for example, right? The idea is that everyone is different because of their genome is different, and therefore they're trying to understand how the genetics can predict what may happen. But the problem and the challenge with that, it's as we just discussed, it's a static book to go from the genome, all the way to a disease or to how healthy somebody may be. It goes through very complex molecular cellular processes, basically, right? And in a certain way, the immune system is one of the mediators of that. And it provides a lot more information because the genome is relatively static. It's static. But what the immune system, as we just discussed, it adapts. It basically absorbs information from the environment and respond and try to maintain homeostasis, right, basically. So therefore, the adaptation and the state of the immune system, where it's at, it depends on time. It depends on the event that has happened. And therefore, it's critical that you track it longitudinally within individuals, right? So that answers that longitudinal question. And the other property is that the immune system, just like the genome, it's very different from person to person. And we're still at the infancy of understanding where those differences come from, how exposure and how genetics, how variables like the microbiome and so on shaped and influenced what we call personal immune states. And I have some more jargon word for that, and we can maybe get to that later. but that actually predisposes us to have different response capacity and trajectory when we move towards the future, right? So in a way, the immune health concept has to do with, can we measure the immune system in a precise way that would tell us what's going to happen in the future to that in that individual, how they respond to vaccines, how they respond to disease, and so on. If another pandemic happens, would this person be competent and capable of responding well to an infection, for example?
Yeah, we went back to the why. But it's fine because when we went to the how, I felt like there was so much more to say about the why. So
the why.
I mean, I'm sold. I'm definitely sold. But one quick follow-up question, just going back to the genome analogy, and it seems like the immunome is sort of the antithesis of the genome in a way, because as static as the genome is exactly how dynamic the immunome is. But there's so many different components to the immune system, right? We've got however many different types of cells, however many different types of receptors. How there's just so much going on. How do you even begin to map it all?
So maybe continuing with the genome analogy. Building on it, because Jess said, and use analogies, right? So the Human Genome Project basically, from its get-go, took 9 years to develop the technologies to sequence the genome. And actually, when it started, it looked like total science fiction. And then it took basically 2.5, 3 years to sequence the entire genome and under the budget expected. And I would say, I guess when John and I were younger, postdoc, around circa 2007, 2010, up to that area, looking at the entirety of the immunome, all the different layers of the immune system, was equally, in humans, was equally science fiction. In fact, it was so science fiction that people basically reverted to working in mice to understand all of immunology. 80% of papers in immunology are in mice, and we much better understand the mouse immune system than we do the human one. And since that time, basically, the community has built the technologies so that it is now totally feasible to measure the immune system of an individual at all the different layers and quite comprehensively. Right, so you're not getting maybe everything, but you're getting very big chunks that will shed light. And not only that, the community has shown that by measuring that information, you can build associations that explain things that you would care about in public health, such as disease associations, predicting survival, treatment responses. It's a huge amount of this because it's dynamic, right? You cannot see in the static genome, but you can see in the dynamic immune system. So the justification for measuring the human immune system, for increasing our understanding, has basically been built in that past 15, 17 years, just like for the Human Genome Project. And the whole idea of the Human Immunome Project is to basically move from from the proof of principle and the kind of original the milestone papers that say that we can do this, to actually make this bring this to the public and to the actionable potential that it has. And for the immune system, that requires mapping it across the globe, because we need to understand variation because we each are so different, right?
Yeah.
So another way of explaining the why, I think a simple one that people would get is that we want to change hospital reference ranges for different things. So just take something like the white blood cell count, right? This is probably the most common or second most common test in clinical labs done today. We've been measuring it in humans since 1957. And it hasn't changed. What has changed pretty much is only that it's been automated. We're still measuring the same 5 white blood cells, so immune cells in your blood. But we actually understand that those 5 are cell types. They're called neutrophils, monocytes, eosinophils, basophils, lymphocytes. Basically are a very, very low level description of hundreds of different cell types, each of which has a different function, each of which the amount of which or the activity of which explains whether you would respond to a treatment or a disease. So getting to a point where we now have the technology to measure this in humans, understand this variation, and then be able to deliver, I think, the next stage of tailored precision medicine.
Can I jump in with just a quick question? So again, just with my data scientist hat on, and I'm so glad, Shai, that you mentioned public health. I think this is such a cool intersection of all of our different fields, actually. I would imagine that in addition to some of the, and pardon my ignorance, but the immunology metrics, right? The certain cell count, all the different things that go into the immune system. Are there other variables? Are there other things that will be documented in this database, like other, demographics or anything else, just to paint this picture? I mean, you're really driving home that all of our immune systems are so diverse and they vary over time. So just curious about some of the other information that might be collected.
Sure. So basically, we strongly believe, it's not just about measuring the immune system. It's measuring the immune system, how it relates to physiology and that environment that John talked about before. So that means, we need to be collecting across the globe. We need to be collecting across lifespan, different ages, different populations, rural and urban. They change your immune system, where you live, what animals you get exposed to. So we need to be collecting all that information. We need to link the blood measurements that we draw from you to measure your cells, your proteins, your T and B cell receptors need to be linked with your clinical status. Did you have flu? What is your cardiovascular health? There's a lot of different questions we can ask to make that connection stronger and better.
Yeah. The other thing I also maybe want to stress a little bit, it's we've been emphasizing the dynamic nature of the immune system, right? But I also wanted to make sure we get across the concept that despite that dynamism, there is a highly individualistic state of the immune system that we have been seeing by these pioneering and human studies that some of us were involved in and been working on it over a decade. So this concept that despite these dynamic changes, different individuals have different sort of set points or starting points. And then these are captured by some of these variables we mentioned, right? Like what kind of cells and what's the state of those cells. Together, they form this concept of a human immune state. And we found that many of these states actually can be temporally stable within an individual, but highly different across individuals. And some of those can persist and stay pretty stable over even years, basically. So we're still at the infancy of understanding how these are shaped and where they come from. And that's precisely why in this project, again, the why, right? We want to collect longitudinal information within a single person and also starting at very early, basically at birth. And then we have different age groups and follow these individuals over time. And also going back to the notion of going global, all of this information that we just discussed and mentioned, they're largely coming from studies of the richer world, like, for example, the US and the European nations. But we have very, very little information for populations like Africa, right? That's 4 billion people right there, for example, right? And then there's also some populations that they've never been looked at, like certain Pacific Islanders. They may provide the relevant natural variation perturbation that allows us to learn something profoundly new, basically, about the immune system. So those are the two aspects I want to sort of get across. It's each one of us have, because of the homeostasis that I mentioned, the immune system is trying to maintain homeostasis. Different people have different homeostatic set points.
Right.
And those can potentially lead to differences in future responses and outcomes. And that's what we want to understand, is to be able to link this kind of set point to physiology, as Shai mentioned, and to the responses and so on. And that's the kind of science that we have the concept of, but we actually don't yet understand across the world.
Okay. So yeah, my next question was going to be, do you guys have any early data to show us? Have you tried doing this in sort of pilot populations at all?
Yeah, of course. Yeah. So those are exactly those types of kind of papers I've mentioned that we and others have been publishing, right? So John mentioned his set point works that have been published and kind of multiple works, and I'm sure he would be happy to tell us more, showing those responses. We've been tracking people longitudinally over time. Since 2007, in collaboration with Mark Davis, who basically launched at Stanford, he launched this study of let's measuring immune health or kind of measuring immune system individuals. And we're still in collaboration, still running this project today. And we've been able, just by tracking the same people now for, close to 17 years, we've been able to see how one person, each, as John said, with their own different set points, how do they change over time? And we realize that actually people change relatively similarly, but at very different rates. Mm-hmm. And their baseline positions different. And so we call this immune age. We're able to assess this, and basically what your immune age score is, which is what is your immune system or your immune system state looks like now when I sampled you today, right? That can actually predict a variety of different diseases and treatment responses. So that's an example. And maybe John, you want to give more?
So we mentioned like responses in a little bit a while ago, right? So we've been using the vaccine as an ethical human sort of perturbation. So you can induce a response, and you know exactly when you give that to some first individual, and then now you can track that over time and look at the response. And based on that kind of study in healthy individuals and also people with various different conditions, we basically saw that depending on their initial set point, where they start before they even receive the perturbation, that provides predictive information on how they respond and how well they respond. And that actually can carry across different perturbations. So the same person may be residing in a certain state, and that actually determines where that person goes. If you give this vaccine or that vaccine, or maybe an infection and so on, right? And then so we've been now tracking, for example, folks at very early stages of life, like 6 months and onwards, and pediatric populations, with the goal of understanding where do these set points come from, how are they being shaped. And we also have done studies where we asked if someone had one bout of infection or inflammatory activation, how would those events change these set points, and then how would it influence future responses. And one thing I think we all have been converging onto, it's initially we typically start with measuring lots of things, right? Like let's say 10 to the 7th, a number of data points per person. So that's what the kind of technology Shai mentioned, right? That we can now measure the immune system well. It's based on these omics and immune profiling technologies. But what we often see, though, is that these predictive information or the state of the system can be captured by relatively small numbers of biomarkers. They may not reflect the mechanism behind the specific types of determinism I mentioned, but they can capture the information about the state of those systems. And so, for example, we have found recently that if you have disease, even different kinds of diseases, divergent perturbations to these individuals resulting in fairly severe disease, despite that the initial initiation point of those diseases being very different, the immune system, in a way, downstream all sends them in very similar ways. You can actually find common response markers. And those same ones, and surprisingly, initially when we saw that, matches quite well with a lot of these aging metrics. For example, the one that Shai mentioned as well, right, previously about aging. And what we found is basically that all kinds of disturbances and stresses, be it coming from disease or coming from even healthy decline in immune health because of aging, they all converge down and are being sort of sensed by the immune system, basically. So despite the high dimensionality of the system, there's a lot of rules and regulation going on, such that there's actually a good chance that we can really understand it, because it's lower dimensional in that regard.
Sarah, did you want to jump in?
No, I'm sold.
All right. We're getting it. I felt like we needed an analogy after John's last thing.
Do you have one for us, Shai? Do you?
No, it's true. Yeah. I mean, like it's a bit of a language barrier here. If you don't speak a little bit of the science lingo, and again, I'm just coming off of 5 days where this was all I was speaking to so many different people. So I hear stuff like omics and longitudinal and just like all of these things. I'm like, yes, I'm with you. I get it. Let's do it. But yeah, let's do an analogy.
Yeah. Well, so I wanted to do the analogy as we switch to talking more about the how, but I'm almost, I do want to say something to people that maybe make a connection here to something I think neither John and I said, and it should be clear. So, we have gone in the past, 15 years, there's a revolution in what's called immunotherapy, right? So this immunotherapy basically means using the immune system as the way to actually to combat the disease by manipulating it with some kind of drug, right? So there are drugs now that allow you to target the immune system and change a particular cell or the immune state. And actually, vaccines are another type of immunotherapy, right? So let's just connect the dots between the fact that we now have drugs that allow us to manipulate the immune system and get it activated, with the fact that basically, what we've been telling you is that the immune system looks different in every one of us. So we have a fixed thing, was a hammer, which is some immunotherapy. And, but everywhere we go to knock, actually it looks different because people look different to one another. So understanding that variation is key to actually understanding who will respond to which treatment. Because the reality is, though we've had a revolution in immunotherapies, we're talking, for the average immunotherapy, it's 20 to 30% of people who respond, right? So choosing the right drug for the right patient is another motivation for this thing. And so that's just to kind of give another on the why, but the how, because I'm mindful of the time. The how is basically, and here's the analogy: we want to replicate McDonald's. so
didn't see
that
coming.
That sounds good. But the field is in a place. So measure your immune system. We have very cool technologies, but in fact, we're not reproducible. Like we measure it and it's very, very costly, and we struggle with reproducibility. So I can measure something in my lab, and I'll call John up and say, "Can you measure it?" It's not necessarily that it'll get the same result, right? And in fact, if you let us scientists roam free, we would probably, if there's such a complex system, we will measure different things, and then we struggle to compare them, right? So the idea here is let's take science, let's take the immunology, which clearly is a field that has value for medicine and kind of understanding, and move it from where the standard unit that people work is the academic lab, right?
Mm-hmm.
Of which has all it's very cool, but has its limitations, to something that's much bigger and a much bigger community. And we think of, I say this is like a CERN for immunology. Like, think about high energy physics, how it moved from just the labs to like something bigger because it needed to. Our research, while being very cool and we enjoy it every day, has reached a glass ceiling without being able to measure at a global scale, without being able to be reproducible. And that requires us to basically set up these sites around the globe that, like McDonald's, and that's the only way I mean it, right? But like McDonald's, are have a set of protocols. They measure, they're reproducible, they generate the same type of data that's comparable, that you can ultimately really build those reference ranges, right? Think about when you go to the doctor and there's the measurement and there's parentheses. They say, is this an outlier? Well, that really isn't reflecting today, the people, your average person. It's parentheses that came from measuring people in the 1950s in a suburb of a U.S. city, but Chicago, I believe. But so that type of, so instead, if you can do this reproducible, if you can produce these site models with McDonald's, you can measure it around the globe in a way that actually is going to be much, we can build these parentheses much more tailored to you. And if we understand how the immune system changes over time, and we understand what your ethnic background is, and where, if you're living in a rural and urban environment, we can make those adjustments. So the how is take the cutting edge science that's done in labs like John's and I's, and make it so that it actually is production scalable ready, and also leverage the correlation that John mentioned before. The fact that when we can measure the different parts of the immune system work together, and we can measure a few of them and learn about a lot of them. So to actually make this cheaper. So the idea of the Human Immunome Project is, let's measure people at the 10 to the 7th type measurements, learn the relationship between them, and reduce it so that next time we're measuring it's only 10 to the 4th. And then do it again, and we lose it down to it's only 1,000 feature. And ultimately, we're able to measure a few things cheaply, but actually tell you a lot about the population.
So may I just maybe quickly add one thing? It's that the key word in that analogy of McDonald's, right? It's the key thing we're looking for. It's something that's replicable,
yes.
Right? So we really want it to be totally replicable, so that you got a site model, and then now it can be that's basically a blueprint, and then now you can replicate it. It's like a cookie.
Yep.
Right. The other thing I think that's also important, maybe back to a little bit of the why, but again, to make it easier to digest, it's the CBC that Shai mentioned. Now you go to your doctor and you got these ranges, right? But the interpretation of those ranges are actually not great. You may sometimes get a report back and say your lymphocyte count, it's this, it's a bit over here, and then now your neutrophil count, it's over here. If you Google or go to WebMD, they give you 25 different potential answers on, this may mean this may mean that, right? So it's not very precise. So where we want to get to it's actually a much more precise predictive. So the word predictive, we want to use that predictive information about what does it mean. Number one, what does it mean for the future, basically. Yeah,
I'm so struck by the fact. Excuse me, by the fact that there are so many applications of this, it's almost like endless. And at both the population level, right, real public health implications, but then also at the individual level. I mean, I know we hear so much about like personalized medicine, will this drug work for me? Like we were talking recently about how there are so many different, antidepressants and we respond differently and some work for, so it's very cool. The applications seem endless. I have to ask, I'm so sorry, this is just the way my brain works, just one logistical question. And Shai, you got me thinking about this with the McDonald's, and I'm thinking like franchising, like, all right, how do we maximize data collection points? Is this like you're doing, like it has to be like prospective collection, or could you like integrate with existing systems, medical systems? I see nodding. So yes, the latter. Is that right?
I think it depends on the specific, because we're very ambitious. We want to cover the entire globe, right? Yeah. So our thinking, of course, would like to make it so that one size fits all. Yeah, right? That's the ideal. But in reality, we do need to deal with localities and specific locals, like location-specific differences. A good example is vaccine. So we haven't talked about that in the how yet. We actually intend to use the vaccine as a probe and a perturbation to. We are not only going to look at individuals at homeostasis, but we want to give them a stress, in this case, a vaccine, and then look at how they respond and then monitor how it's almost a test, basically, right? But ideally, we want to give exactly the same vaccine to everyone in our study. But the reality is that's unlikely going to be the case, because some region of the world, they may not get seasonal influenza vaccination, for example. But maybe another vaccine would benefit them better, and maybe another vaccine will be more informative. Therefore, that's something that we need to factor in our design. And the second, I think what you're alluding to is there may have already have been cohorts ongoing and maybe samples bank, and can we take advantage of those? Yeah. And the answer is yes, as long as it fits in with our specific study goals and we can apply the approach to those in a way that we see that the outcome and the data coming out of it will allow us to now getting to the AI a little bit now, to allow us to train models to do this kind of predictions. We will be very excited to incorporate those as well.
Yeah. But yet to just add, there are no real sizable cohorts that have actually been doing this to date. So if you look at the cohorts, there are cohorts that are very large but actually collecting very little on people, or you have cohorts that are small and collect a lot. So there's we strongly believe in partnering here, and this is such an ambitious goal. You cannot do this alone. As I said, it's a CERN for immunology and for everybody coming in. But it could be that bringing in that old data that people have is not worth the while to the extent that we would wish it was.
Understood.
Right. I was also more referring to like existing longitudinal cohort that people have been tracking, but they may not have been collecting the right kind of sample that we need. Right. And we can really partner and work together on that, basically, right? And so here's a good example. There's a program called All of Us in the U.S., funded by the NIH. The goal is to look at 1 million Americans across the country, right? And the goal was initially largely a genetics project. They want to look at their genomes and then link those to all sorts of other variables in health and disease in these individuals. And to study the immune system, you need to look at immune cells in addition to other things like circulating proteins and molecules. But at the time, it was logistically not that easy, actually, to really get the blood and process them in the right time and so on, so that they can collect the kind of cells that we want to look at, for example. So that's an example of a program where it's very large, but the information that one can generate from the samples they have collected so far, at least in most of the people, it's difficult to do what we aim to do. So therefore, we need to either add in additional sample collection into cohorts like that, or for certain areas, regions in the world, we need to build them from scratch.
Understood.
Did we answer the how?
I think we did. And by the way, we are double the time. We were like, we'll record for 20 minutes. We're at almost 40. So I don't know, Sarah, do we want to...
we did the discussion.
We did
one
a
of those
lot.
papers where the discussion is mixed with the...
We jumped around.
We did jump around.
Yeah, you tell the student, what is this writing? Go back and clear it up.
No, really, this is incredible, Shai and John. Really, thank you both so much for joining us. As you said, it is incredibly ambitious. My mind is I'm a project management type brain, so I'm like, oh my goodness, there are a lot of steps that have to happen here. But really, the applications are incredible and endless. Sarah, I don't know if you have any final thoughts or anything before you wrap up.
Best of luck, honestly. I'm sold on the why. I'm honestly a little skeptical about the scope of things, but I mean that's all good science, right? You got to aim for the stars.
So I'll give you an analogy. The part, right?
Another analogy.
So the I think the reason you say, the why is cool, and that we told you there's evidence our labs can do this, and really we're talking about scale. And I think the reason it's difficult for people to think about this in many ways is because academics are kind of, that's the thing we suck in, right? But right. I was on a flight recently, and I sat next to a guy who does very large real estate and construction projects, and I explained this Human Immunome Project, and then he said, well, what's the budget? I said, it's in the low billions. To do all of this, and there's coordination. And he said, large construction projects, right? That like we did the tunnel in San Diego, that was $6 billion, right? So I think, it's always the comparative field. And so it just shows that this is not a project just for the academics. We need the people who have the project manager, the production, the scaling, all of those need to be coming in to do something, but ultimately, the complex, and that's how you solve these complex problems. Yes. And we drive on them every day.
We're not trained to think like this. We're trained to think small.
And physicists, they have demonstrated this to be one way in which they can manage very ambitious large projects, right? So if you remember the LIGO detector that won a Nobel Prize for detecting gravitational waves, for example, right? So that took huge teams of people spread around the world, and initially came from a conception from theoretical physicists. And at the time, it was like, who's going to implement such a detector? That sounds like crazy and expensive, right? And when you also look at the scale of the cost, like Shai mentioned, a few billion. Let me ask you a question. Do you know what's the 1 year annual budget of, let's say, just one institute at the NIAID? They're responsible for allergy and infectious diseases. Do you know their annual budget?
No clue.
Not
It's sizable. It's 7 or so billion 1 year, right? So 1 year they spend about $7 billion to support allergy and infectious disease research around the country and the world. And so that's how we have all these vaccines, and it's amazing. But when you think about the kind of why we want to do this and the kind of question we can answer and the applications, and then you ask, should we invest $3 billion into something like this? I hope the answer is yes.
You have our votes, that's for sure. Tag us in if you need any help. Clearly, you need our help. Yes. No, that was a joke. No, really, what you're doing is amazing. Thank you so much. This was really an incredible discussion. Sarah, do you want to take us home?
Yeah, thanks so much for tuning in, you guys. We hope you learned a thing or two. I certainly did. Please join us next week on your trusted source for no nonsense, just science.